Okay. So when we say mutation, mutation means once they can inherit from one generation to next generation of cell, one generation to next generation of the cell. Okay, that cell could be anything. It could be ovum, it could be sperm. Okay, that is different story. Okay, but when we say changes means mutation, because mutation could be two type: inheritable means which can move from my generation to my child's. Okay, that is different. Okay, but when we say mutation means there is a somatic mutations also took place, which will move from like skin cancer from one generation to other generation of the cell. Okay, so that's why I'm telling inheritable changes in the DNA structure of a cell, and which will move from one generation to other generations of the cell cycle. In general, what will happen? Mutation are harmful. In general, mutations are harmful, either neutral. In general, mutations are either harmful or neutral. In our case, if you talk about, okay, means human, maximum of the mutation is neutral. They don't have any impact. The reason why they don't have much impact, because we have 98.5% of our genome that is non-coding genome. Okay, so that's why they are hidden in the non-coding sequences. That's why we don't have maximum of the cases, we don't have any impact. But if there is other mutation, in general they are going to have the harmful effect. Okay, very rare mutations are there, which is fruitful, and that is the cause of the diversity. That is the cause of the diversity in the any of the species are in genus. Okay, so when we talk about evolution, okay, evolution concept work on the mutation only. Okay, due to the mutation, many of the species survive, many of the species they finished. The reason is these mutations they will help for the adaptation to the environment. So if we got any mutation which is not making us adaptive to the environment, we will be lost. Okay, but if some mutation will take place and which will cause us to adapt to the system, then obviously we will survive for longer phase of the time. Okay, on the earth. Okay, so that's the thing here. Okay, now come to the types of DNA mutation. DNA mutation is basically divided into three sections: genome, chromosomal, and gene. When we say genome mutation, now I'm talking about human mutation only. So when we talk about the genome mutation, means when whole set is increased, like rather than 2n, you have 3n, means rather than 23 pair, 23 triplet, 23 quadruplet is present. That is known as genome wide mutation, polyploidy. Aneuploidy means I plus 1 or minus 1, means either 47 or 45 chromosome, either 45 or 47 chromosome. So aneuploidy is, many books says it is genome wide mutation, some books say it is chromosomal mutation. Okay, different book, different way of characteristic. Okay, so what will happen? Aneuploidy is an important aspect which can survive, but not all time. Deletion of the chromosome is more dangerous than the addition of the chromosome. If you have added extra chromosome, there is a chance of survival, but deletion, that is very less chances of survival. Only the case Turner syndrome. In the case of Turner syndrome, what will happen? One X chromosome is deleted, but still they survive. But if it is the autosome is deleted, chance of survival is zero. Okay, they generally discarded during the fetal development itself. Okay, second one is chromosomal mutation. Means if there is any changes takes place in chromosome structure, means some section have been duplicated, or some section have been deleted or some section have been translocated to the other chromosome. So that is known as the chromosomal mutation. Gene mutation, if there is any mutation takes place inside a gene, that is known as the gene mutation. Here, so genome, as I said, triploidy, tetraploidy, okay? But triploidy is most common out of all polyploidy, okay? How these ploidies takes place? How it is possible to have the triploidy in any fetal development? Any one of you? You all have studied cell division in the cell and molecular biology. So tell me, it is somewhere related to meiosis. Tell me how it is possible to have the polyploidy. Hmm? How it is possible to have the triploidy? Means you haven't listened the lecture very carefully yesterday. Hmm? It is generally due to the non-disjunction or improper segregation. It is when in what will happen meiosis, okay? So meiosis one, what will happen? One chromosome set, they come closer to each other, and then only one side kinetochore here kinetochore express, and they will segregate it like this one. Okay, so what will happen from that 46 chromosome? They will have 23. Now, this 23 chromosome they will double, means they will present as a chromatids. Now, both side, one of them, both side the kinetochore will be developed. Okay, so they will be segregated both of them. So by that way, we get 23 in the meiosis. But imagine during the process of the first meiosis, while they have to just express one side of one chromosome, but they express both sides, then what will happen? At this stage, chromatid is separated. Okay, so what will happen? They will have after meiosis one, one side they may have the 46 chromosomes or chromatids. So what will happen? 46 will be 46. So what will happen? It is just double number in that. Either it could be sperm or ovum. Okay. So the process is known as the non-disjunction, improper non-disjunction or segregation of the chromosomes. Okay, during the meiosis. If you don't know this process, go ahead, read again about meiosis. Okay, aneuploidy is same concept, but rather than having all the chromosome will start expressing both side kinetochore, only any one of them they will start to doing it. That's how one of the cell may have n plus 1, other will have n minus 1. Now this n plus 1 or n minus 1 whichsoever will fuse with the ovum or just vice versa. Okay, then what will happen? Either they may have 45 or 47 chromosome. Okay, so that is known as the aneuploidy. Yeah. Now come to the chromosomal mutation. It is rearrangement. Okay, like here deletion. This section have been deleted. Extra chromosome, we talk aneuploidy. Here duplication. This section is duplicated. Yeah. Inversion, flip-flop movement. Okay, flip-flop movement. So what will happen here if you will see inversion? They are blue, brown here, but what will happen? Blue, brown is moved. Okay, then translocation. this section have been taken up from the other chromosome and some section of this chromosome have been shifted to the other chromosome, but not in same pair of chromosome. Always remember, translocation is not same pair, not chromosome 1 to 1. Okay, between one type of chromosome to other type, means chromosome 1 between chromosome 20, chromosome 20 versus chromosome 22, like that way. Okay, but not in same chromosome. Clear? So that is known as the translocation. Okay, now gene mutation is various type. Okay, it could be base substitution, means one nucleotide that is substituted. Okay, insertion and deletion, one or few one or few nucleotide added or deleted. Okay, dynamic mutation means where depurination will take place, or some of the changes in the structure of the nucleotide takes place, that is known as the dynamic mutation. So how it is possible to have all those type of the substitution or anything? Generally, it can take place due to the replication error. Sorry, but the rate of error is very low. The rate of error is low. How much base pairs we have? Human have how many base pairs? You can unmute and tell me. Only millions is not going to work. Exact number. Three billion. Billion is million in 10 to power 6, 10 to power 8. No. 3.2 10 to power 9. 3.2 10 to power 9 base pairs we have. Okay. So what will happen if you will talk about that bases only, then it is just double, 6.4 10 to power 9. So what will happen during the process of replication? If you remember, I told you the chances of error is 1 in 10 to power 9 bases added. So every mutation you have a chance of 3.2. Every replication process, 3.2 bases could be mismatched. Okay, means you can say 3 to 4 bases can be mismatched every time. Okay, so obviously it is a small quantity, but yes, still it is there for mutation. Second most important cause is the repair enzyme defect. Means if the repair enzyme didn't work properly, then only they may have the defect. Okay, now come to the base substitution. Base substitution could be two types: transition and transversion. Transition means same group, means pyrimidine exchange with pyrimidine, purine exchange with the purine. So what will happen is structurally same, so they can be more adaptable. Okay, they can be more adjustable. Okay, so what will happen, that is much easier and it's more prevalent and more common is transition. When we say transversion, it is one group of nucleotide changes to the another group, means purine to pyrimidine or pyrimidine to purine. Okay, but transversion is rare than the transition. Okay, now once these substitution takes place, in the coding area, first I'm talking about now coding area, forget about non-coding area. In the non-coding area, generally they will not have any impact. Generally I use the word. Okay, I haven't said they will never ever have the effect. Okay, generally the mutation don't have impact on the non-coding sequences. Okay, but imagine if the mutation means the base substitution takes place in the coding sequence, then what will happen? Okay, in that time what will happen, there is three type of mutations: silent, missense, and nonsense. Three types. Silent mutation means if they got mutated, but ultimately they are synthesizing same amino acid. If you remember about the codons, what codon says, that three letter, okay, these three letters they code for an amino acid. So if you remember for glycine, proline, you have seen there are several codons are there, okay. So what will happen if one mutation takes place and they change the codon, but still that codon code for same amino acid? So what will happen? The protein structure will not change. If protein structure will not change means there is no mutation take place. Ultimately to the protein means effective molecule don't have any mutation. Okay. Next one come to the nonsense mutation. If any type of the base substitution will takes place, and this base substitution will cause the changes to the stop codon. If a base changes takes place, okay, and that will causes the base to the codon to convert into the stop codon. So what will happen due to that reason? Truncated protein will synthesize. Truncated means small size. Truncated means digested, or you can say small size. Okay, so a truncated protein will synthesize. Now brush up your knowledge from your high school, okay, and whosoever went for the undergrad, okay, because I can see the faces, maximum of them they are past the high school, okay. So what will happen when we say nonsense mutation? It didn't mean that always you are going to synthesize the useless protein. Your high school books, many of the undergrad books says once there is a nonsense mutation takes place, you will synthesize a truncated protein which is useless and it should be discarded. That book says many of them. Okay, undergrad book and the high school books. But what I'm telling, listen carefully. It's not the case. It's not the case. Only we can say we will synthesize a truncated protein. It could have no activity or reduced activity. We don't know. Sometimes they will have no activity at all. In that case, obviously they are useless, they have to be discarded. But in several cases it have been found, find they have the reduced activity. Means still they have some activity, but reduced activity. How it is possible? Think of a protein. You now know the protein. This is the protein structure. Okay. This is the one active site. This is the other active site for a single protein. Means two of the enzymatic activity is present in this whole protein. Now imagine a mutation causes this finger. Okay. This finger have not been synthesized, but others are synthesized. So what will happen? Active site is still there. Only what will happen due to the absence of this finger, okay, they may have less stability. Maybe possible previously they have their lifespan is 5 minutes and now their lifespan may be 3 minutes. So if lifespan is reduced, means what will happen? They will survive for less time, so they will have less activity. Clear? So it's not like always you will say stop codon will always synthesize. No, not always synthesize. They always only synthesize one thing, truncated protein. Always synthesize truncated protein, but truncated protein is not always the inactive protein. They always synthesize truncated protein, but the truncated protein is not always inactive protein or not the useless protein. They may have reduced activity. Clear, everyone? So now come to the missense mutation. Okay, missense mutation can be two types, conservative and non-conservative. Okay, if they are conservative means they are replaced by one amino acid to other amino acid, but same group. Same group means hydrophobic to hydrophobic, acidic to acidic, basic to basic, hydroxyl group containing to hydroxyl group containing. Clear? Same group, same group. Then there is a high chance, I'm using the word, there is a high chance that this protein may have their functionality and structural similarity. If it is conservative, there is a very high chance that they maintain their three-dimensional structure, and if they maintain their three-dimensional structure, they will maintain their activity. Okay. Once we talk about the non-conservative, non-conservative means one group is changed to other group, means acidic to hydrophobic, acidic to basic. Okay, this type of changes means one group of amino acid change to the other group. Then there is a high chance, then conformation of that protein structure will change. Then there is a very high chance that conformation of the protein structure will change. This conformational change may cause, again I'm using the word, this conformational changes may cause the protein dysfunction or inactivity of the protein, or reduced activity of this protein, or a novel activity of a protein. Okay, so again I'm not saying always they will synthesize the useless, no, I said conformation change will take place due to non-conservative. Okay, this conformation change may cause inactivity of the protein, reduced activity of the protein, or a novel activity of the protein. Okay, something new activity will come. Okay, that is known as the non-conservative. Okay, like sickle cell anemia case. Okay, so there what will happen, rather than glutamate, valine comes. So glutamate is a which type of amino acid? Anyone? As per classification, glutamate is a which type of amino acid? Okay, polar, non-polar, but tell me if it is hydrophobic, acidic, basic, hydroxyl group containing. Okay. Okay. What does it mean by alpha? Unmute yourself. Talk to me. It is a discussion. It is not a lecture, basically. Okay. So tell me, which type of amino acid is a glutamate? You all are going next term, MD1. You are forgetting these points. In MD1, you will have minimum, I'm telling, in biochemistry and genetics, 4 times course, 4-5 times extra. How you will survive then? Whatever we are teaching in biochemistry whole term, okay, you will finish in first 3.5 weeks. Whatever you've gone through in whole term, you will finish in 4 weeks. That's it. So imagine the course is 15 weeks. So minimum 3 times extra work. Same thing with genetics. And you are forgetting what is the glutamate, aspartate. Even the name, you all are going to organic chemistry. Okay? ATE. If any group have ATE, means it is acidic group. Glutamate, always remember like that, glutamic acid. Aspartate, aspartic acid. So in chemistry, that's why what is requirement? That you must know organic chemistry. Okay, so whenever acidic group is there in any of the compound, what they used to add either acidic or at the last ate, 8. Okay, you ate the molecule. Okay, so aspartate or glutamate, it is a acidic. Now valine is which group of amino acid? Glycine, valine, aliphatic. Very good. In the aliphatic, it is a non-polar. Anything else? Valine have some unique characteristic. If it is a branch chain or not?
Branch chain.
Very good. It is a branch chain amino acid. So, if you change from acidic to non-polar, and even branch chain, so that will cause the conformation change. Okay, in the beta chain of the hemoglobin. Okay, so due to that reason, they got a novel property also. So this novel property causes in beta chain a surface with the hydrophobic amino acid. So what will happen when they are present in the uh, deoxygenated form, means oxygen is not available in that erythrocyte. So these are many of the beta chain, they will attach to each other due to this hydrophobic interaction. So they make a rod shape structure. This rod shape structure causing the distortion in the structure of the erythrocyte, and that is the sickle cell anemia. Clear? So that is caused by the non-conservative type. Okay. Now other type of base substitution, RNA processing mutation, means it is non-coding area now. So cap site, if it is there, or poly A site or splice site, what will happen? If cap site means, then ribosomes cannot attach. If polyadenylation site, it will take place. Then what is the use of poly A site? Tell me. What is the use of poly A? Any messenger RNA? Transcription is going to the part of the final exam. Always the worst result in the pre-med program is genetics, I'm telling you. Always. And whosoever haven't got B plus in genetics, they always struggle in the MD program. I'm telling you. So, because why you don't care, it is 2 credit hour. Less number of lecture, but it is more complicated than the biochemistry. I'm telling you. So poly A not for stabilization, something else. It guides something. Stabilization only in the case of stabilization means it reduce the chances of the degradation. Which type of degradation? If exonuclease will come, they will not cut down the messenger RNA structure. Okay, so yes, obviously it provide a type of stability. Okay, but along with that poly A tail, that will guide them to come out from the nuclear pore. Okay, so they help them to come out from the nuclear pore. Okay, they are guiding. So if poly A tail, it is got mutation, then what will happen? That guiding force will not be there. So what will happen? They are hitting to the wall, nuclear membrane. So what will happen? Whatever their lifespan is there to synthesize the protein, that will be over inside the nucleus, or even they come out. Till that time their lifespan is over, so they will be degrading. Okay, splice site mutation will be there. Some intron cannot be removed out. If intron cannot be removed out, unusual long messenger RNA will synthesize. Unusual messenger RNA, unusual protein will synthesize. Okay, so even that's why I told you, in non-coding sequences in general they don't have problem. But here you could see these all are present in the non-coding area, but still they cause the mutation. Okay. Regulatory site mutation means Tata box, GC box. Okay. So these places, if mutation will take place, the binding of the RNA polymerases that will be affected. Okay. Yeah. You could see what are the effect. Okay. Sequences and their effect. Okay. Now come to the insertion and deletion. Means point deletion or point insertion. If a single nucleotide base pair have been added or deleted. So what will happen? The reading frame will be changed. If you added one, then what will happen? They read always three bases. Okay, so whole reading frame will change. That's why it is known as frame shift mutation. Okay, if one is deleted, reading frame will move opposite direction, smaller section. Again, frame shift mutation. If two bases are added or deleted, again frame shift mutation. Only the case if three bases are added or three bases are deleted, then the impact will be less. Because what will happen if three bases are deleted or added, one reading frame is added or deleted, then the chances are there they will synthesize still some protein, but the worst case is the frame shift mutation rather than, even I'm telling, stop codon. Okay, insertion of stop codon, or once you will say nonsense mutation, it is worse than nonsense mutation, frame shift mutation. Okay, same time insertion deletion could be large insertion and deletion means rather than one or two bases, a huge number of bases can be added or deleted. That happens in hemophilia B. Okay, so line. Okay, large interspread segment nucleotide that will be added or deleted. So line and sign is repetitive sequences. Okay, so rather than sign, line will be added if any time or deleted. Okay, in the hemophilia. Okay, so that is the cause of hemophilia. Okay, so this is also a type of mutation. Yeah, if you will see, this is missense nonsense frame shift. One base added and that causes whole changes. Means basically they synthesize totally different proteins. Here you will see base pair insertion or deletion. Okay, here stop is there means what will happen? It is a nonsense. Okay, here frame shift totally change. Okay, so that's why I'm telling totally change is more dangerous than even stop codon. Okay, now dynamic mutation means where due to some reason a dynamic changes take place, like trinucleotide repeats. Okay, many genes are there where in their coding or non-coding sequence there a repeat of the trinucleotide takes place. Repeat of trinucleotide takes place. So what will happen? It has been observed several times it has been observed that when RNA polymerase, or not RNA, DNA polymerase, when they are going for replication, sometime they will become more sluggish or relaxed during that process. And anyhow, once they are going through, these DNA will go up rather than tightly attached to each other. They will be like this one. And they keep synthesizing. Means rather than imagine there are 50 trinucleotide repeats are there. They will synthesize 52, 53. So what will happen? Next generation, they will have more. That is the reason of a diseases. A lot of diseases takes place due to the trinucleotide repeat. So it is belongs to the dynamic mutation. Other dynamic mutation, if you go through, like here, these are the some of the cases are given. Like in the exam, if there is trinucleotide Hutchinson disease, regulatory region, Fragile X syndrome, Fragile X tremor also takes place, myotonic dystrophy takes place, Frederick ataxia takes place, different type of disorders are there. Okay, here you could see. So here if you will see name, which type of the repeats are there, if you will see. Okay, where they are present. Okay, where they are present. What is the usual number of the trinucleotide repeat and number of repeat when they will have the disease, this one. Okay, so these are maximum of these disease, either they will affect the neurons or skeletal muscle, maximum of them. Okay, yeah. Somatic versus germline mutation. So what will happen? Somatic versus germline. Somatic mutation means in the cell. Yeah, okay, which will go from one cell generation to next cell generation. Okay, when we say genetic or germline means the mutation takes place in the sperm or ovum or when they are developing, fetal development during that time takes place. Imagine a mutation takes place in 16 cell stage and one cell got the mutation. Now all the tissues which is going to synthesize by this cell, whatsoever tissue is going to synthesize by this cell, they all will be having mutation, while other tissues which is synthesized by other 15 of the cells, they will have different type of means normal DNA. So what will happen? They will not have severe impact, but mild impact of mutation they will have. So time of mutation is very important. Time of mutation during the fetal development is very important. Imagine if a sperm got mutation, then sperm and ovum fuse, all the cells will have. Then what will happen? Severe sign and symptoms they will have. If it will take place two cell stage, one of them they got mutated, other one is not, then 50-50% cells. If 8 cell stage, 1/8, 16 cell stage, 32 cell stage, depending upon which stage that will take place. Okay, if any of the animal or human have two type of genome in same body system, two type of genome means one part is mutated, other is normal. Okay, that is known as mosaicism. That is known as the mosaicism, means like mosaic you have seen in flooring. Okay, in flooring what will happen? If you will add different type of stone, different color, that is mosaic. So same word is used, mosaicism, if two type of genome is present in same individual. All females, all females are mosaic. All females are mosaic. The reason, either they will have this X active or this X active, but at a time in a cell both X cannot be active. Either this one will be inactive or this one could be inactive. So that's how what will happen. Some of the cells have this type of, some of the cells have this type. Okay, so that's why females are always known as the mosaic. Okay, next one come to the here. Then DNA damage. Other way of the damage means again it is basically due to the failure of the repair damage or error during the damage. Means once you are repairing and that time rather than recognizing something proper, it is not properly recognized. So what can cause the induced mutation? Induced means which will activate the chances. Internal metabolic processes, depurination, demethylation, deamination are different type of chemicals, carcinogens, mutagens, teratogens. Okay, they are causing the induced mutation that will causes the DNA damage. Okay, so causes of mutation is spontaneous and non-spontaneous. Spontaneous means due to many reasons like mistakes in the DNA polymerase or during the segregation or some chemical product, means like we are taking water and all those things, that will cause us the spontaneous changes. Like here, spontaneous aberration, aberrant segregation, error in DNA replication, toxic metabolic product in our body system itself. Induced mutation due to the presence of something like mutagen, carcinogen, teratogens. Okay, so they will changes the structure, means they activate. So if anyone can tell me what is the difference between three terminology: mutagens, carcinogens, and teratogens. Unmute yourself. Talk. Okay. Tell me. Hmm?
Carcinogens cause cancer, mutagens cause mutations in DNA proteins formation. And I
Yes.
think teratogens is for, if anything, causes like not an error, but like anything negative to happen to a developing fetus or an embryo or something.
Very good. Very good. Very good. All three you know very clearly. So teratogen means if any type of mutagen, it is again mutagens, okay, which may cause the defect in fetal development. Okay, means fetus development is affected, that is known as teratogen. Okay, like vitamin A. Vitamin A also behave as a teratogen. If excess vitamin A is used during the pregnancy, it behave as a teratogen. Okay, not natural. I'm not talking about natural. I'm talking about supplement. Okay, vitamin A is also teratogen. Okay, mutagens means any type of mutation. It may cause cancer, may not cause the cancer. If it is causing the cancer, it will known as carcinogens. Because mutation, any type of mutation, but all mutation don't cause the cancer. So that's how these three terminologies there. Carcinogen, type of mutation which will cause the cancer. Teratogen, type of mutation which can cause the development defect in the fetal. Okay, mutagens, any mutation which can cause the changes in DNA structure. Okay, now come to the spontaneous. These are spontaneous means deamination are different type of the group. Okay, red oxidizing damage, hydrolytic damage, uncontrolled methylation here. So depurination, due to the even water, okay, purines are deleted here. Okay, so what will happen if purines are deleted? Let me make it increase size. Okay, here if you will see here, deleted. Okay, depurination takes place here. Okay, here you will see during the process here one purine is deleted, so this will cause the apurinetic site. Okay, now once if it will take place just before the replication process, so one strand, this one, second strand will be this one here. Okay, so if anything synthesized from this strand, that will be normal, but whatever the DNA synthesized from this strand, then what will happen? They will not have this one. So what will happen? Either this base can be missing, means one overall missing base is there, means that could be cause of the frame shift mutation. Okay, or they will try to repair, but they don't know what they can be adding anything. So this is known as the spontaneous mutation. If you will see here, apunic here, 10,000 purines lost every 20 hours. Means in a day around 12,000 mutation is just by the depurination. Okay, then spontaneous mutation, deamination. Amine group is removed out from the cytosine. So if deamination of the cytosine takes place, they will synthesize the uracil. Here if you will see this structure, this amine is removed out, so in place of that oxygen will come, means it is the ketonic group you can say, or you can say phenolic group, not phenolic. What is this name? When double bond O is there, OH is phenol, ketone, ketonic group I think they say, na? I don't know. Anyone? You people are going organic chemistry. If it is a benzene ring and double bond O is there, what they used to say? Ketone? Anyone of you? I know phenol is there when hydroxyl group is there. Then it is phenolic group. No carboxyl group. Phenoxide. You all are using those terminology I don't remember. Not aldehyde. Aldehyde is anything. CHO group is there. But a specific name should be there. Okay, I will ask Dr. Audin. I really don't remember at present. It should be ketonic group. Let me check what is ketone. Okay. Ketone again, it is, I'm just looking if
It won't be ketone because the
ketone
double bond.
is normal. Ketone is anything could be there, na? Means
No,
even
there has to be an extra carbon for it to be a ketonic group, I think.
okay, let me benzene ketonic. What they say that one? The name I want to know. Ketonic. I can't. Phenol I know, but aromatic compounds. OH group.
Is it benzenoaldehyde?
Really don't know. I'm searching.
I think
it's
that is
old.
benzenoaldehyde.
Benzylaldehyde. Okay. I'm looking. I can't find even in Google quickly. Hmm. Okay, I will ask Dr. Odin. It's been long, means rather than long, I will say ages. When I studied the organic chemistry, I can't say own hexane, own. I don't know how to name it. Okay, I will check. I will check later because I have studied the organic chemistry in last time 1999. It is 21 year before. Okay, so really I remember phenol, but I really forgot what is they will say ketonic group when it is added. Okay, so here if you will see in this PowerPoint, what will happen here? Ketonic group like something is added. Okay, so what will happen? This one is known as uracil. So uracil is not part of the uracil is not the part of the DNA. So they will be easily recognized by the enzyme DNA glycosylases and they will be repaired. That's okay, very easier. But the problem comes if methyl group, 5-methyl cytosine. Okay, so 5-methyl cytosine actually what will happen once they will got the deamination, they will convert into thymine. Remember, this is very important point. When first they will cytosine get the methylated, and after methylation, they will get for the deamination. Then they will make the thymine. Now, one side you have guanine, one side you have thymine. Then it is known as the hot spot for mutation. Means it is 50-50% chance rather than the cytosine should come. They may change the guanine to the adenine. Okay, so here you could see depurination, deamination. Here, depurinated may be possible they are removed out, and then what will happen? Frame shift mutation means one base is deleted. Here, in general, if they are using this one, they will be repaired, no problem here. Okay, here. Next one is the spontaneous or means tautomeric shift. So here, if you will see, this is the keto form, okay, which is present, common form, but they can convert into the enolic form, okay, due to the tautomeric shift, okay. So what will happen once these changes take place, means keto to enolic form, amine to imino form, okay. So what will happen rather than ATGC composition, they have the ACGT composition. Okay, they will have ACGT composition rather than ATGC. Here you will see ACGT base pair are formed. So imagine again if they got changed tautomeric shift just before the replication process. So where A is there and it goes the tautomeric shift. So rather than having T, they will make the C. Then again, this is a mutation. Okay, so that is the biggest problem takes place in the tautomeric shift. Okay, induced mutation due to some chemical, like here, many of the chemicals are there and which causes the changes in the DNA structure. Okay, next one is thymine dimerization. So in this dimerization, what will happen? DNAs are attached to each other like this one. Okay, but here what will happen on same strand, one strand, two of the thymine they attach to each other. Rather than here, they are attached to each other here. Okay, so what will happen? Rather than making bond between the two strands, same strand, two of the thymines are attached. Okay, that will cause a slightly change, and in general these changes are repaired. There are two type of enzymes are there. One is the photolyase enzyme. Photolyase enzyme, once you come out from the beach and stay at home, after some time photolyase enzyme, they will cut it down between here, the bond, and again they will attach to each other. But if it is not repaired, that could be the cause of the skin cancer. Okay, clear? So this is the thymine dimerization, and it is very common. It is very common in the case of the uh, those who goes to the beach and all those places. Okay, so that is known as the thymine dimerization. Okay, now come to the here mutation rate. Leave it, it is not very important for you, but just try to understand likelihood that a gene will be altered by a new mutation. Okay, so what will happen? A gene like expressed as a number of new mutation in a given gene per generation, like hexokinase gene. What are the chances? Okay, so it vary between a strain of the same species, means bacterial. Okay, while when say mutation frequency, the chances of a mutant allele in a population. So generally, mutation frequency is a constant number. It is don't change quickly, but mutation rate can change quickly. But these things are not important for you now. Come to the DNA repair, means how they are repaired. If they are not repaired, like here, thymine dimerization, they may cause the skin cancer. Okay, so type of direct repair means, try to understand this concept. When we say direct repair, means backbone is not affected. This backbone is not changed at all. Okay, whatever, like deep urination takes place, remove that one and add. But you are not breaking the backbone. That is known as direct mutation. Clear? Next one is the base excision mutation. Backbone is broken down, but only one nucleotide is removed out and added. One nucleotide is removed out, added. New one. That is the base excision. When we say nucleotide excision, more than 10 bases, they are cut it down and resynthesized. That is known as the nucleotide excision. Then there is the fourth one, recombinational repair. Recombinational repair, if a long set of the DNA that got mutated and they will not realize, then they will go just like if you remember of the prophase 1 of the meiosis, where they will go for crossing over and they will go for the resynthesis. Okay, same way recombinational repair takes place. They will use the second set of the DNA, means chromosome 1 to 1. Okay, they will come parallel to each other and they resynthesize this section of the chromosome. Okay, that is known as recombinational repair. Okay, yeah. So you don't require to remember these enzymes, just try to read one time so you will understand. There are several of these enzymes, they are basically utilized for the replication process. So what will happen once you remove something, then you require something of them, they have to be resynthesized. Like here, DNA glycosylases enzyme. These are group of enzymes, they are very good enzymes which will recognize the abnormality in the DNA structure. DNA repair and diseases, many of the diseases, these are the diseases and what are the phenotype and enzyme which is affected. Okay, you don't require to remember any one of them, just I'm giving an example. Okay, so come to the direct repair, like thymidine repair. Here, thymine dimerization, take it, you just cut it down. So which enzyme? Photolyase enzyme. Here you could see photolyase enzyme. Okay, so they will help to cut it down again, repair it. So that is known as the direct repair. Base excision, as I told you, immediately just one nucleotide, like cytosine to uracil, due to the deamination process. But if you will see 5-methyl cytosine, they will convert into thymine. This is the hot spot. Okay, they cannot be repaired, they don't know which one is correct. Okay, so base excision, as I said, it is one nucleotide. Here you could see one nucleotide is removed out and added new one, repair. That is base excision. Nucleotide excision means when more than 2-3 bases are hampered, like the thymine dimerization. If photolyase cannot do it, okay, if photolyase cannot do it, more than 10, around 30 base pair, around 30 bases that will be nicked out, means they will cut it down from here to here and then resynthesis. Okay, that is known as the nucleotide excision. Here you could see they cut it down and resynthesize. So that is known as the nucleotide excision repair enzyme. Okay, so we are done with this lecture. Okay, so let me stop the record.